So first of all, open the NCBI database. Search for the gene sequence for hemoglobin. Filter it out by selecting the genomic and protein coding sequences. Select Homo sapiens from the right-hand side to get information regarding hemoglobin in humans. After that, you will see that results got filtered out and everything related to only hemoglobin in humans are shown. As hemoglobin has several different subunits, you can see details associated with each of them. From those, I will retrieve the sequence for alpha subunit only. After selecting it, you can see that sequence as well as other information related to alpha subunit of human hemoglobin is given. From this section, select the FASTA sequence tab. Here you will see the entire gene sequence of hemoglobin alpha subunit and other details associated with the sequencing data. Copy and select the sequence data and paste it in Notepad. Once data is saved, let's begin with primer generation. Start by copying about 18 to 30 bases from the beginning of the gene sequence. Afterwards, open NEB Tm calculator. Select Taq polymerase from the preference section and paste the nucleotide sequence for forward primer in the first box. Copy last 18 to 30 base from the gene sequence. Paste it in the second box of TM calculator. You can see the finalized report and change the primer sequences accordingly. Always remember to keep the primer sequence length near to 18 base length, in such a way that annealing temperature of both the primers is around appropriate range. After copying the sequences, open Reverse Complement tool. Paste the second sequence in the dialog box and submit it. Now copy the reverse complement sequence to the notepad and you got both the forward as well as reverse primer sequences.